bright field. This is a picture of the cell under white light. Then we have the fluorescence signal, the creatine transporter here, and a dye for the endoplasmic reticulum. And so the point of this is to get quantitative information about how much these mutant transporters are trapped inside the cell. And so basically for each one of these images, pixel by pixel, you can measure how much yellow there is and how much purple there is. And then basically when you plot those two things against each other, you can see how highly they're correlated. And so that gives you a similarity score for each image of the cells. So here's some of the cells expressing normal creatine transporter. You can see this little halo of protein at the plasma membrane. You can see some of the more severe mutants, the proteins trapped inside the cell. And so this is a distribution of several thousand single cell measurements. And you can see similarity between the ER dye and the creatine transporter is fairly low for wild type. For the mutants that cause misfolding, you see this really high similarity indicating the proteins, again, trapped inside the cell. And then you have mutants that are somewhere in between. So again, the point of this is just to see, if there are different types of defects that are caused. So we did this for all 10 variants. And then to get another sort of quantitative number, we did another type of flow cytometry experiment where we stained the plasma membrane, the surface creatine transporter with one fluorescent antibody, and then we stained the intracellular creatine transporter that's stuck inside the cell with a different color. And so you can see these contours show you, again, several thousand single cell measurements. And you can see basically, again, you have these severe mutants where there's no protein at the plasma membrane, but it's all intracellular, right? Then you have the ones like wild type where you get a little bit of both. And then you have some that are in between. And so if you just turn these numbers into sort of the percentage of protein that's at the surface of the cell versus inside the cell, you can see basically about 32% of the wild type protein makes it to the surface of the cell and pumps creatine. Our neutral variant looks pretty similar, but most of these, they vary a lot, but most of them have some impairment of transport to the cell surface. Okay, the biggest thing that the net outcome that you're looking at, right, is how much creatine do these various mutants pump, right? So we basically did an uptake assay so we could sort of compare that to the extent of intracellular retention. And you can see basically here's the wild type protein. Just look at the gray bars. You can see we get about 3 picomoles under this condition. You get a similar amount for the neutral variant, but the one thing that all these mutants have in common is you lose the net ability to transport creatine, right? That makes sense. If you compare these numbers to the plasma membrane protein numbers and the ER retention numbers, you can start to see some interesting patterns. So for most of these mutants, the amount of creatine uptake that's lost, that's on this axis, sort of scales with the amounts of protein that makes it to the surface of the cell. Then there's a couple of them where you see a little bit of deviation, and then a couple where this doesn't fit at all. So these two mutants, G421R and P382L, they get to the surface of the cell just fine, but you still lose creatine uptake. So these mutations are actually probably perturbing the structure. And so if we go back to our model of the creatine transporter, what you'll see is it makes a lot of sense actually. So creatine transporter, when it pumps, basically it has this outward facing conformations. It goes back and forth to pump creatine. And in between that cycle, this lid basically clamps down and closes the transporter. And the first residue that we see that impairs function basically should should make it more difficult for the transporter to close. This one that has a deletion of this phenylalanine 315. I talked about that in the beginning. And this basically makes a direct contact with creatine when it's bound inside the transporter. So deletion of this, again, would interfere with the ability of the transporter to hold creatine. So again, we can see how these mutations cause a spectrum of molecular defects. If you put this all together, what you see, we have our ER retention, we have the amount of protein in the plasma membrane, we have trafficking efficiency, and then the relative creatine uptake. And you'll see basically most of these mutations shown here in red, so-called class, basically lose protein at the plasma membrane, right? So you have 80–90% reduction in the amount of protein at the membrane, and that explains why you don't get creatine uptake, right? But there's a second class where, in this case, your reduction is pretty subtle, right? 20% reduction in the amount of protein, but a complete loss of function, right? These are ones, class 4 variants we call them, that actually cripple the ability of the transporter to do its job. And then you have some that are in between, like these two, where you get a pretty significant reduction in surface protein, but you know, the reduction in surface protein can't account for the reduction in function. So there's probably a little bit of this, a little bit of that. And so, as I'll talk about this in a minute, this actually can be really valuable information when you're doing screening and when you're targeting clinical trials, if you're trying to fix the broken transporter. And towards that goal, one of the things we want to look at is whether some of these mutants could be rescued, at least in this context, right? So one of the classical things that you do for misfolded protein is drop the temperature of the cells down to about room temperature. And you see if you can rescue the transport of the protein in the plasma membrane. And you can see for two of these variants, we can more than double the amount of protein in the plasma membrane. And for one of these variants, G132V, that drop in temperature actually does translate to more creatine pumping. And that's actually really interesting, because basically what it says is the protein's trapped in the ER, but if you can get it to the surface of the cell, it will do its job. That's not true for all variants, but it's true for this one. The other thing we looked at was this molecule 4-phenylbutyrate, which I think the Vienna group's going to talk about tomorrow. And so this is actually, I believe it's a clinically approved molecule, and it basically rewires the ability of the cell to fold proteins. And so I think they had published a paper that suggested for certain mutants, you do actually see a rescue of the expression at the surface of the cell. We looked, and for these particular variants, we don't really see too much change in either cell surface trafficking or in function. So it's important, I want to say our results in no way contradict theirs. The methods are different, the mutants are different. Everything I see suggests that our findings are pretty well aligned. But I think our results basically suggest that 4-phenylbutyrate is going to be targeted. It's going to be maybe good for some mutants, but not others. One thing I will say that's interesting is we do see a significant uptick in the function of the wild type protein in the context of the 4-phenylbutyrate. And that actually could be interesting for people that are thinking about clinical trials, because the female patients with CTD should have haploinsufficiency. They should have one wild type copy. So it's possible that this compound might help rescue or boost the activity of the one wild type allele. So just something to keep in mind. Again, not clinical advice, but just some interesting food for thought. Now, why does this matter? Why do we care about what the mutants do? Why is this potentially interesting from a drug development standpoint? Again, I want to circle back to cystic fibrosis. And for those of you, who might be not so familiar with the story, right? Actually, Vertex Pharmaceuticals has gotten three or four compounds through clinical trials in the last 5 years, and it's completely changed the treatment of this disease. But to get there, right, they spent, the community spent 30 years figuring out how to do this. And what they ended up doing was screening for two different classes of molecules. One type of molecule they call a corrector, which basically helps get the protein from the inside of the cell to the surface of the cell. And the other is so-called potentiator, which basically increases the activity of the transporter at the surface of the cell. And you can see sort of why this would be important, because if the protein's inside the cell, it doesn't really matter if it's more active, because it's not going to be at the membrane where it belongs, right? To do this, basically, they came up, they had to figure out what the mutants do, and they had to screen against the appropriate mutants. And they actually ended up taking that information and targeting those medications in clinical trials to the specific patients with specific genotypes. Now, here, it's clear that you don't have a huge number of patients, and that might be a little complicated. But in terms of screening, this is still useful, because you could, for instance, screen against 382L or 315. These are mutants that get to the surface of the cell just fine. And then you could sort of screen for molecules that increase transport once, once the protein's where it needs to be. And then I can imagine, again, this 132V variant, right? It's inside the cell, but if it gets to the surface, it's functional. So if you screen your test compounds against this variant, you could potentially identify compounds that increase transport of the protein, rather, in a way that you could distinguish between sort of the effects of the compound on trafficking versus function. So again, I mean, it's a different disease. It's a different scale, right? They have different tools. So these things aren't directly translatable, I don't think. But I think that you could sort of see this as a template if you wanted to find molecules that fix broken transporters. So I should note, our lab doesn't actually have funding for this right now, so I'm not sure how far we're going to go with this. But if anybody's interested, we have a lot of things we'd be happy to share with anybody who wants. We have a ton of genetic constructs for transient expression. GFP tag constructs, HA tag constructs, mutant constructs. We have stable HEK cell lines that inducibly express these transporters from a defined locus. We have structural models of the transporter that are confirmations that out. We also are working parallel. And so we're doing that for cystic fibrosis and retinosis. We could hypothetically do Marty Salazar. He did most of this work. He works. He took a master's and is currently working in pharma. This is Wes Penn. He did a lot of the cloning. Chuck Kuntz and my collaborator Yuli Kohler at Simons Foundation did a lot of the molecular modeling and did a great job with that. And yeah, so I'd be happy to, I guess questions will come later, but that's basically it.
Thanks so much, Jonathan. That was